biochemistry at Biogen University of Technology. After I came to Biogen Institute, I have the knowledge of coding before. So I'm now thinking, how can I incorporate it into the biochemistry knowledge I've been studied in school? But by getting here, I realized that is being achieved. Also, in school, most of our biochemistry deal with molecules. When I said molecules, I mean carbohydrates, proteins, and all that. But when I come here, the applied bioinformatics in design vaccine and drugs, and also in making of diagnostic kits. Before, I thought vaccine design and diagnostic kits, they are the big things. But by coming here, they break down into a simple that I can take in and understand. So that has helped me to shape my knowledge about chemistry that I've been learning and coding that I've been learning since. They are not just about building websites and reading about proteins. It can be used in design vaccine, making diagnostic kits, and also design more tools through my coding skills.
My name is Elajide Elizabeth Oluwatobuju, 400-level student of biochemistry, Biogen University of Technology, Ogbomoso, Oyo State, Nigeria. I'm here at Biogen Institute for my industrial training, and I've gained a lot of experience in medical research, bioinformatics, and genomics. And I've been able to use some of the bioinformatics tools in vaccine design, construct, and diagnostic kits. I think I've learned a lot, and I think it's really been my career path.
My name is Taiwo. I'm a graduate of veterinary medicine from the University of Biogen, Nigeria. I came to Helix to get more skilled in biomedical research, to learn the applications of the tools we learned in school to real life science. I've been in Helix for this period. I've been able to learn the application of bioinformatics, genomics, transcriptomics, and some other biological concepts in designing humanity, like vaccine designing, drug design. Another way in which my time in Helix has helped is that it has helped to broaden my scope as regards my career opportunities after school. So before I came in, I only had a limited idea of what I could do with my certificates and the knowledge I gained in school. But coming here to Helix has helped me to broaden my scope of what I can do after school. Now I can be a force for global change.
My name is Victoria Oluwatosin, studying biochemistry at the Biogen University of Technology. My stay here has actually given me more insight into my career. Before I came here, there is this feeling of what will I do after school? The exact aspect that I'm venturing to after graduating as a biochemist. But getting NSLG Institute has helped me to navigate on several opportunities that lies ahead of me as a biochemist. We were taught more of theories in school, so when I get here, we were taught more of practicals, like we really personalize what we do here, and it has really been a good experience so far. I'll be able to gain more, to learn more about bioinformatics and also life science in general. So I'll be able to know more about how to provide a solution to problems of the public health, like in form of research and bioinformatics in particular.
My name is Ochai Blessing. I study medical laboratory sciences from University of Jos. I'm a graduate. One of basic necessities of every human is being able to access quality and affordable health care. But far from that, aside the quality and affordable health quality, let me dwell on that a little bit. I don't think quality can be achieved when people don't have the right knowledge to be able to give that quality out. Helix is a place that has afforded me this opportunity. They've been able to bridge a gap for me. In fact, a very important one. Prior to this time, I have just been exposed basically to bench work and theories. But my coming here has exposed me to the computational aspect of biomedical science. So it has bridged that gap. I've been able to understand that, oh wow, there is another aspect that has to do with the molecular aspect of life science. And as a matter of fact, the molecular aspect of life science is what really give expressions to some of the illnesses that we get to have. So if I don't get to understand what's really happening at that level, I wouldn't know how to be able to attack it or to address it better still. So Helix has given me that opportunity. Doing genomics, bioinformatics, and all of that, it has really exposed me. I don't feel that in life we stumble on things. I feel that everything is pre-programmed for us. So it's a matter of you, setting yourself in the right position at the right time to be able to take up such opportunities. And I feel this one is an amazing one for me, and I'm glad I took it up.
Okay, Malone.
Yeah, good morning, everyone. It's a great pleasure to have all of us here this morning. And I want to believe everyone is hearing me. Can you hear me, please? If you can hear me, please, can you just say hi? Yeah, thank you, Dr. Emeka Igwe. Yeah, great. I'm so happy that we can hear me loud and clear. Perfect. Yeah, we promised to start by 10. I'm just trying to wait a little bit for others to join. Yeah, let's just give like 5 minutes and let's listen to one of our videos for the next 5 minutes, and from there we kickstart. Who is that? Okay. Voice ẹ̀ náà jáde, fídíò jáde.
My name is Ochai Blessing. I study medical laboratory sciences from University of Jos. I'm a graduate. And one of the basic necessities of every human is being able to access quality and affordable healthcare. But far from that, aside the quality and affordable health quality, let me dwell on that a little bit. I don't think quality can be achieved when people don't have the right knowledge to be able to give that quality out. Helix is a place that has afforded me this opportunity. They've been able to bridge a gap for me. In fact, a very important one. Prior to this time, I have just been exposed basically to bench work and theories. But my coming here has exposed me to the computational aspect of biomedical science. So it has bridged that gap. I've been able to aspect that has to do with the molecular aspect of life science. And as a matter of fact, the molecular aspect of life
Um, we need to start now, and we welcome each one of us to this training. Uh, it's just an introductory telling us what bioinformatics entails and what is capable of doing in the community of science and what we can actually use it for at this time of today. And first of all, I want to thank all the participants for joining this call for us to actually learn and see what we can gain from it. From the video that we have shared with us, just to share a few testimonies of what is happening here at Helix. And my name is Dr. Oladipo Kolawole, the founder of Helix Biogen Institute, which is a transitional research hub where we build capacity in the aspect of biomedical sciences. And we are sited in Oyo State in a secondary city called Ogbomoso. And that's where we are. We have a quite number of students in the lab working. We take students on IT, we take students on internship, and this has really allowed our students to acquire necessary skills that have landed some of them in US, UK, Australia, Canada, and even some have even gotten some international jobs based on the skills they have acquired. One of our major focus in Nigeria and Africa at large is capacity building, looking at the background that some of us had, that we emanated from a low background. And that is why we are trying to domesticate science, domesticate bioinformatics in Nigerian, among Nigerian youth, so that it can be a leverage to improve and find solution to ourselves. So I welcome each one of us. We're going to run our first hour today, 10 to 11. Then tomorrow we'll run our 2 hours where we'll be doing some hands-on. So two of us will be taking us this morning. We take us introduction to bioinformatics. Then one of my team members to will take us through some of these things. And actually, in early two, we have our last space for people that want to do their work in molecular biology and biomedical sciences. So we help people to sequence at a very affordable price if your project needs sequencing. So that is what we do. And now, as a serve as consultant for anyone that want to start a startup in biomedical. So we have a lot of part of services that we render at Helix Biogen Institute. So to start with, you are all welcome. God bless you. Like we said, participating in this training is very, very, it's very, very free. It's only if you just need certificate to support it that we requested for token. So if you don't pay, it doesn't mean you can't attend the class. So you are free to attend the class. And those who have paid for the certificate, that use the Paystack link. By tomorrow, at the end of the class, you will have access to your certificate immediately. Those who did clinical bioinformatics can testify that that same day they have their certificate once they paid to the Paystack platform that was provided. So the class will end tomorrow. Immediately tomorrow, we want to send all our certificate to us for those who actually are interested and have made some token of the payment requested from you. So I think we need to kick start. Let's move to our microphones and let's be attentive to what we have. So let me share my screen. Can everyone see my screen? Perfect.
Yes. Yes.
Great. That's good.
Yes.
So I can go to sharing mode so I can start that. Now, I'll be introducing us to bioinformatics. And what is bioinformatics? I want to use a very simple language. It's just a marriage of biology and information. We all know what is called biology. That's the study of life and information technology. So it's one of the recent developed science using information to understand biological phenomena. And when we talk about bioinformatics, it's also involve the computational tools, methods used in managing, analyzing, and manipulating volume of biological data. Now, bioinformatics can be regarded as part of computational biology. And when we talk about computational biology, it's the application and quantitative analytical techniques in modeling and showing problems in biological system. Also, bioinformatics is an interdisciplinary field, acquiring advanced knowledge of computer, mathematics, statistical methods in order to understand some biological phenomena at the molecular level. It's not that new. It has been introduced since 1990s, and it deals with the management and analysis of data pertaining to DNA, RNA, and protein sequences. Although it may include some other biological data sets. Such as gene expression profile for those who are studying cancer. When we talk about RNA-seq, and we have for protein structure too. We have protein interaction, we have microarray, even have about functional analysis of biomolecules. We have what we call in drug design. Now, bioinformatics cover a very broad range of area. One of it is functional genomics. That is just identification of genes and their respective functions. So we do that. We can use bioinformatics to infer the functions of a gene. We have what we call structural genomics. That is prediction of structure of protein and elucidating the properties, what is capable of doing, and other things. Another one is also comparative genomics, understanding the genomics of different species of organism or even diverse species of organism to see the similarities and differences that are shared so that we can have a scientifically inferred functions. I've talked about microarray. It's a level of gene expression in tissues. For instance, taking histopathology, looking at cancer cells, and look at the various stage of development in different kind of diseases. We have medical informatics too, which involve the management of biomedical data, which is special reference to biomolecules and vitro assays and clinical trial. Now, what are the components of bioinformatics? We have creation of databases, which involved, and a database is just like a bank that stored and management of biological data sets in an organized manner. And database can be accessible. You submit a new one, We have a lot of database. After this class, somebody will be taking us through databases. And it's so unfortunate for now, Africa do not have any database. We do submit to what is database hosted by US, NCBI, or ENA by Europe, or that of Japan. Now, in bioinformatics too, you can develop algorithm of statistics, and that is development of tool. And I'm happy to tell you that by the end of this year, Helix Biogen Institute is launching a bioinformatics tool that was developed here in Nigeria, developed here in-house. And we have really validated it very perfectly. It's already sent out for publication, but the training will come out by the end of this year. And it's going to be free for everybody to use because that's the way it's done in the Western world. We're just trying to let ourselves know that Africans can also do something in the African soil that is useful for everyone. And it allows you to compare protein sequences, data already existing. In analysis of data, you can analyze your DNA, you can analyze your RNA, you can analyze your protein sequence, you can analyze your protein structure, you can analyze the gene expression profile, and even biochemical pathway. Like I was sharing someone, we have a tool that you can even know the glycolytic pathway your organism is using to degrade a certain compound. For those who did petroleum course of chemical engineering or microbiology, that their organism will degrade a certain thing, but they can't even know the pathway by which the degrading is occurred. Bioinformatics tool can allow you to do that by God's grace. And what do you need as a bioinformatician? It's just an international computer network. You need a group of computers that can communicate, usually where it exchange data between users. Then you need an internet. Then once you have an internet, you have a laptop, you're good to go as a bioinformatician. Then which area have you also applied bioinformatics? We're using in sequence mapping of biomolecules. We use identification of nucleotide sequences for functional genes, finding of sites that can be caught by precision enzyme. For those who are into CAPS or CRISPR technology, bioinformatics have been of help. Then we have prediction of functional gene products. Then it has helped us to trace evolutionary trees of genes. Look at the era of COVID, SARS-CoV-2, we're able to trace where it emanated from Wuhan. Evolutionary tree really helped us in that. You have molecular modeling, you can model your molecules, you can design drugs, you use bioinformatics to design vaccine. And that is what we have done in Helix. And God has given us the grace to make the name because one of the candidate vaccines that we designed for COVID-19 was captured by WHO on their list as small as Helix Biogen Institute is. So like I do say on our platform, everything we are teaching you from the point of practical is what we are doing, we are teaching. What we don't know how to do, we don't teach. And just chipping in, we just almost running up an intern, a virtual intern for some of our students, in which some other one will still come up, will be getting you involved in it. That are going to do for bacterial bioinformatics. That's for 3 months, weekend only, because of those who are working and academic staff that want to be acquire the skill so that it can fit into their schedule. We have for bacterial bioinformatics coming, metagenomics coming, molecular docking coming, and mRNA vaccine design coming. The only problem we actually have, we have a limited slot for each of those class. So it's help us to develop model for functional tissues and every other thing. So these are the things that bioinformatics has really helped us to do. So one thing we are able on in early, it's not about the theory. we have done the theoretical work in the university, but here is about skill acquisition. So I will be calling on one of my team members, Mr. Pulu, to take us through biological databases. It's from there that we're going to see how to go about bioinformatics. I think I have two people raising up their hands. I just want to take those two people. Is at the end of the class that I would like to take question. Khadijat Isha, do you have any question? I see raising up your hand. Khadijat.
Sorry, it was a mistake.
Okay, cis. I don't know cis. I don't know what it means. Now, Mr. Bolu, are you there?
Yes, sir. I'm there, sir.
Yeah, I think you have the floor to take us through databases.
All right, sir. So I would like to share our screen. Good morning, everyone. Please, can you hear me clearly from your hand?
Yeah, we can hear you. Good morning.
All right. So please let me quickly share my screen so that we can comment. So let me try to share my screen with us. So I believe you can see my screen now. So
Yes, sir.
okay. All right. So thank you very much. I will quickly run us through introduction what biological database entails. So by tomorrow, by God's grace, we're going to have a little hands-on how to retrieve data from some of the database that will be introduced to us today. So today, I just want to do a quick overview, introduction rather, to what biological database entails. So for some of us, I believe we must have had one or two knowledge about what biological database entails, and majority of us must have attended one or two seminars, maybe did one or two courses online just to get yourself acquainted with this particular line of research bioinformatics. So I would just like us to kindly follow through as I put us through this particular session. So biological database. So what is biological database in the recent? So from here, let me just quickly run us through what bioinformatics is. I believe my Dr. Ladipo has already given us a little clue about the definition of what bioinformatics is. So from my hand, just talking from the aspect of biological database, I would like to like this to define bioinformatics now. Bioinformatics is the application of information technology to store, organize, and analyze the vast amount of biological data which is made available in form of sequences and structures of protein. That is, as we all know, that protein is the building block of any living organism, and also we also have nucleic acid, which is responsible for carrying of information from parent to their offspring. So also these two form in which these two forms in which data exists now, the protein and sequences now, there's a particular form in which their data exists in them. So for example, for the nucleic acid now, sequences, data are always available in the form of sequences. So we only have sequences of nucleic acids, and also for proteins, we have two forms in which the data of protein exist in online databases. One of it is it exists as a sequences and also as a structure. So you as an individual, if you don't have interest in working with the sequences of protein now, you can probably work with the structures of protein, and we have diverse databases that archive or store structures of proteins, which you can easily assess, and then from there you can start to work in start whatsoever project that you might probably want to involve yourself in. So now, how does biological database? What is biological database, and how does it came into being? Biological database is a computerized archive used to store and organize data in such a way that information can be retrieved easily via a varieties of search criteria, and databases are composed of computer hardware and software, basically for data management. That is just one of the definitions for biological database, and also it can also be said to be a collection of data that is organized so that the content can easily be accessed, managed, and updated. So one objective or major reason why databases were created is just that people could have access to data, and also for proper management of data, and also updating of data as knowledge increases. So this is one of the reasons why researchers have to come together to just to just develop a particular space whereby several of these data that are being generated from different researches can be stored. So that is just it. And how does biological database emerge? Biological database emerged as a response to the huge data generated by low-cost DNA sequencing technology. Now, sometimes back, we had little understanding of what DNA sequencing entails, but lately, DNA sequencing technology known as being the latest things in terms of use to authenticate your wet lab analysis, or to validate. Several of your work is just to uh, you carry out your project at the molecular level, and this entails you working with genetic information of your organisms. So, and from this, we can easily give meanings to uh, the uh, the the processes that occur uh, within the biological system. So that is just that is just how biological database came into being. And one of the first databases that emerged during those time was GenBank. And this GenBank, now I will be talking about GenBank, collection of all available proteins and DNA sequences. And this database now is maintained by the National Institute of Health, the NIH, and also the National Center for Biotechnology Information, NCBI. And one thing that actually paved way for the creation of data of GenBank is that it paved way for the Human Genome Project. I believe some of us must have had an idea about what Human Genome Project is. And Human Genome Project is just it allows complete sequencing and reading of the genetic blueprint of man. And from this particular project, we're able to understand uh, the genetic informations that are present in the entire human being, right from the soul of the crown of the head to the sole of the feet. That is just what Human Genome Project helps us to achieve. And from this particular project, after the whole project was completed, then the data was stored in a database that was created, which happened to be GenBank today. And from there, today now, we can access different information from the GenBank. So from this particular project now, we all can have ideas to the various variations that cut across different human races, talking about the single nucleotide polymorphism. Maybe you're talking about why a particular disease condition is so much predominant in a particular group of group lies. One of this particular project now gave us ideas into those questions and the likes. So now the data that are stored in biological databases now is organ of optimal analysis, and it consists of two types. We have a raw data, and we also have annotated data. So a raw data now are the primary data that are generated from the research work, and also we have annotated or curated data, which are secondary data that are worked on by researchers by researchers who don't have the capacity to develop or to generate their own personal data in the course of their analysis. That is just and the data that are submitted now to biological databases now are basically are meant for indexing, optimization, and data organization and optimization. As we all know that optimization is just design and operation of a system or process just to make it as good as possible and such that it can be defined in a good sense. And talking about indexing now, it helps us to arrange this data in alphabetical order, listing them in uh, based alphabetical order and also based on their locations. So also we have some basic features of biological databases, which I have itemized on my screen here. One of it is that a biological databases is complex. It's complex, such that we have different types of data that are being stored on databases. So this is just what actually makes it complex. And also due to this complexity now, the data that we have there now is heterogeneous in nature. It's not just a one-way data. Some databases can have us both sequences, structures, even several other informations in regards to a particular data or a particular project that is being carried out. So because of the heterogeneity now, also it is dynamic. We have the data that we have on databases now keep on changing. At the beginning, when I was trying to give the definition of what database is, I said one of the major functions uh, why database had to be created is just was to update latest information. And due to this update now, data keep changing on the database. So that is just why it is dynamic in nature. And because of this dynamic now, uh, several data kept seems to be inconsistent. So inconsistent. So can you see my screen? Please, can you still see my screen? So, sorry, can you see my screen from your hand? All right. Thank you very much. Thank you very much. So let's proceed. Okay, sorry. I'm still currently on this particular screen, so that's the reason why I have not. So when by the time I'm done with it, I will move to the next slide. Thank you very much. So now, and also databases. One other features of biological databases is that it has high high volume of data, and also the one of another feature is that for data sharing, that is ability to just to not just to store alone, but also sharing the data that are being generated with other researchers who doesn't have access to them, and also for data curation. So now, what are the objectives of biological databases? So we have, as I say on my screen here, I have four different objectives of biological databases, and one of it is that it helps to make all relevant data available at one place. So that is just what biological database help us to achieve. So it means all the data that are relevant data that are similar, that are of the same project, now that are similar to each other now, it makes them available at a particular place. That is just what it helps us now. And also, and due to this, it has helped us to ease the stress of trying to like traveling before we can get a particular data set to execute our project. So that is just it. And also, another objective of biological database is that it helps to make biological data available to the scientists. So not just only for data storage alone, but also making it available so that so that people can have access to it and they can use it to execute their project. There are some databases now that also beyond storage of data, they can also provide tools that can help to analyze or interpret uh, our data. So that is that is just it. And also another objective is to help update existing information easily. So once there are new information regarding a particular data set, there is a need for the management of a database now to uh, help upload them on their database so that people can have idea as to what is currently in vogue in line of that particular data or research work. So now, why do we have to access data? So the summary of the reason why we have to access data now is just basically on uh, comparison of our data with what has already been in existence. That is just one of the that's just a summary of everything that why we access data. So some of us must have had idea of what blasting and this blasting multiple sequence element and the likes. Those are several ways by which we can compare our data to what has already been in existence on other databases, and we have different tools. We have softwares and online tools that can be used to compare our data with what has already been in existence. So that is just the summary of the reason why we have to like access biological data. So what are the usefulness of biological databases? Number one, it helps the researcher to study available data and form immune disease, antivirus, helpful bacteria, and medicine. So this is just one of the usefulness of biological databases now. By the time we have access to a data, from there we can draw inferences and arrive at a conclusion. And even in the process of our research projects now, we can come up with different innovations that can help to solve diverse uh, human problems in terms of their health and the like. So just like my boss have said, God has helped us in this institute, and we've been able to come up with a particular candidate vaccine that can help to combat the SARS-CoV-2 infection, which is COVID-19. So it's as a result of the data that we archive from all the databases that we use. We worked on them, and from there we're able to come up with this particular candidate vaccine. So this is just one of the reasons why biological databases is useful in present day biomedical or life science researches. Also, it helps scientists to understand the concept of biological phenomena. One of the reasons why bioinformatics came into being earlier on was the medical scientists, the medical doctors, the medical lab scientists. Now, majority of them, one thing, the issue they had then that they were not able to understand the basic processes that occur in the biological system, and this actually gave them a lot of headache. One thing that actually the problem they had at the inception was that how to isolate DNA, and from there, due to knowledge, they were able to come up with processes and procedures that can be used to isolate DNAs from human and from of several organisms, uh, and from there, by the time work were basically done on this particular organisms, on this particular data now, it was there that they were able to understand how biological processes take places the human uh, in the human. We're able to understand basic processes that occurs in our system, such that if you as a scientist you have interest in designing a vaccine, one of the steps that is involved in designing a vaccine is we carried out immune simulation, and immune simulation is just one of the ways by which we test run if the candidate the construct we are designing now actually uh, is actually going to stimulate an immunological response in our body system, and from there we and at what rate is this particular construct going to be initiating the immune responses, and at, uh, also it also gives us the duration of time in which these antibodies will be produced in our body system. So these are the one of the ways by which we get understanding on the norms. If we were to inject our test sample into rats, mice, and the likes, into lab animals now, we can only see the production of antibodies, but we might probably not have ideas as to the reason behind the production of those antibodies. But with bioinformatics now, we've been able to analyze and give meanings to the process to the uh, the processes behind the functionality or the production of different antibodies that are produced due to vaccination. Due to vaccination. So also, uh, it can also give us ideas as to why a particular test sample, a particular construct can have effect on a particular organism. Take for example, you are trying to come up with an antibiotics that can help to inhibit the growth of particular microorganisms. Now, so we do molecular docking a lot for some of us that are interested in drug design and computational biology. Most times, what they do most is just they do molecular docking and molecular dynamics simulation just to have ideas as to the interaction between the biomolecules that are involved in the construct. Also, one usefulness of biological databases is the database acts as a storage of information. So it helps us to store information and also helps to remove the redundancy of data. So now, what are the types of biological databases that we have? Now we have different criteria in which we use in classifying or categorizing our databases, biological databases into different groups. One, on the basis of the nature of data, we have different types of database under here. One, we have the structured database. Two, we have, and in front we have examples of databases that store structures uh, of structures of proteins. We have the PDB, which is the protein database. We have the scope, and we have the scope, the cat. Also, we have the protein database now, and examples. We have the sweet spot. We have the protein information resources. We have the Tremble, the translated in European molecular biology laboratory. Also, we have the ProSite, and under that, we also have the nucleic acid database. And these are databases where nucleic acid can easily be accessed. Example of them is the GenBank, the DNA Data Bank of Japan, the EMBO, EBI, and lice. And also, the second criteria that we can use to classify our biological databases into different groups is also on the basis of the sources, that is the source of data, the sources in which you are getting your data from. So in one, we have primary database, and we also have second database in on the based on these particular categories. And the third category we can use to classify our biological databases into groups now is based on the accessibility, how often we have access to data on this database. And in this slide, we have particular two types of database. We have the public database, which are databases that you as a scientist, you don't need to pay a token or to register before you can access data for them. And also we have private database, and these are databases where we all have to you have to pay a token or you register with them before you can have access to the database. Most times, maybe you are trying to they may probably help you to analyze your data. Maybe you are trying to predict the structure of your and maybe you want to interpret the result of your analysis and the likes. So these are the two types of database under this particular criteria. And one thing that is sure is this: the examples I gave in the first I gave above now. Some of these examples can fall under each of the uh, each of the types of database that we have, independent of the basis of the criteria that we use in categorizing them. For example, the PDB, for example, is a structured database, can also be a primary database on the basis of the sources, and can also be a public database because you don't need to register on the PDB RCSB database before you can access data from them. So also the Swiss plot also being a protein database on the basis of the nature of data can also be a primary database and can also be a public database. Now for ProSite is a protein database based on the basis of the nature of data, but it's a secondary database, and also it's a public database. So these are just, these are examples can fall on any part of the of the group of databases, type of database that we have. So these are just some of the logos of different databases that we have. So now let's just in the course of this class, I will major more on the two types of database that we have based on the basis of the sources of data that we have. So now one, we have the primary database. So now what are primary database and second database? This is what we are going to be discussing, and from there we move to other database that we have, the composer database, and from there we call it a day, so that by tomorrow we are going to have hands-on in on this particular course. So now, what are primary database? These are they are known as archival databases, and basically they contain experimentally derived datasets such as nucleotides and protein sequences, as well as structural informations of macromolecules. So now, this particular database, what they do is uh, they archive data that are in their raw state. These are data that are generated from the laboratory, from wet lab, and they are stored on the database. And basic informations can be accompanied by functional annotations and bibliographies and linked to other databases. So now, by the time you are submitting the data now on this database, there are some information that is needed or that is required of all of the scientist or the researcher now to okay, sorry, Miss Ibukun, it seems you are raising up your hand, please. Miss Ibukun, what on? Can see you raising up your hand. Okay. So now I said there are some information that is required of the researcher or the scientist that is trying to submit such data online. And the essence of this is uh, such that the management of this database now can look for other data that are in relation to what you are about to submit, so that they can put them on the database. That is just to reduce the redundancy of data. That is just what they help us to do. And now the data to this database now are directly submitted by researchers themselves. And once a data is submitted, the data is assigned an accession number, which is permanent and become a part of scientific record. And we have different examples of primary database. We have the NCBI, we have the ENA. When I'm talking about NCBI now, I'm still referring to the Gen Bank. And also, we have the ENA, we have the DNA Data Bank of Japan, we have the GUs, we have the AEA, we have the Swiss Prot, we have the PDB, we have the PIR. So now, these are examples of the databases that we have on the basis of their nature. So now for the NCBI, the ENA, the DDBJs, I'm still talking about the nucleotide database. I'm talking about the GU. GU is a gene expression omnibus. This particular database now helps to archive or store data that are in relation to gene expressions. So maybe take for example, you have interest in downloading you have interest in working on a particular gene expression. Maybe take for example sickle cell anemia. So now you're trying to like look at the expression level in a patient. So now the one of the best database to visit is the GEO database on the NCBI, which can easily give you access to several data in that particular line of research. That is just it. And also likewise as AEA. For the Swiss Prot, protein databases where we can retrieve the sequences of protein. And also for PDB, PDB now is a database, primary database where we can have access to structures of data. And also if you're looking for information regarding to a particular data now, protein data now, PIR is one of the best database that you can access to give you information in that particular line of data. So we also have some databases now that are publicly accessed by researchers. That these three databases now, they are the major databases that researchers across the globe often have access to retrieve their data. And this particular three databases now, they came together to form an acronym called the International Nucleotide Sequence Database Collaboration (INSDC). And what are these databases that form these particular acronyms that come together to form this particular association? We have the DNA Data Bank of Japan, we have the NCBI, and also we have the EMBO EBI. So now the NCBI reside in the US, the NCBI reside in America, while the EMBO EBI reside in Europe, the DDBJ also reside in Asia. So now these three databases now they came together to share information among themselves so that to prevent any form of conflict among researchers, such that everybody can have access to the same information on these three databases. Although one thing that actually that differentiates the information that are being retrieved from this database to be unique now is that the way in which each of the database now stores their data differs. So the accession number of data on NCBI cannot be used to access the data on the EMBO API. Neither can the accession number of the data on EMBO API can be used to access data from the DNA Data Bank of Japan. So each of these databases now has their own unique accession number that can be used to access data from them. Among these three databases now, both NCBI and EBI provide a portals which allow researchers to have access to several other databases now that are embedded in them with a single query. So now, by the time you input your search query on the NCBI or EBI now, you can have access to several other primary databases that are embedded on the database. So that is just it. So now we have the National Center for Biotechnology Information, the GenBank, and this particular database now is the NIA Genetic Sequence Database. It contains an annotated collection of all publicly available DNA sequences and part of the Institute of the INSDC. And this particular database now updates information on a regular basis, approximately every 2 months. And we have several divisions within this particular database. So this is just the interface for the NCBI database. By tomorrow, I will be teaching us on how we best can retrieve data from this particular database, and I will also be giving meanings to some of the link that we have on this database, how best we can utilize them to carry out our research work. So now the next database I will be considering is the EMBO EBI. So it is the meaning is the European Molecular Biology Laboratory, that is EMBO, and this particular database is a subsidiary of the European Nucleotide Archive. So by tomorrow, by the time we want to do the hands-on on this particular database now, we are going to navigate our path through the European Nucleotide Archive ENB database to the EMBO EBI. So to just for us to see, because virtually all the information that we have on the EMBO EBI now are managed by the ENB. So now, and EMBO EBI is just a comprehensive database of DNA and RNA sequences, and EMBO nucleotide sequences now database is collected from scientific literatures, patient offices, and is directly submitted by researchers. And this particular database now has been prepared in collaboration with the GenBank and also the DNA Data Bank of Japan. That is, it is part of the INSDC. This particular database now is established in 1980 and is maintained by the EBI, the European Bioinformatics Institute. So now this is the old interface of the EMBO EBI. So by tomorrow, by the time we are navigating our path, into, or we try to explore this particular database now, the interface has already changed. So there's a new update on the interface itself. So now the last database now, another database that we have is that make up the INSDC is the DNA Data Bank of Japan. It is the only nucleotide sequence database that is located in Asia, and this database now, 75% of the data that are stored on this database are generated from Japan, and followed by Asia and several other countries that are present in the Asia continent. And this database was established in 1986 by the Center for Information Biology under the National Institute of Genetics of Japan in collaboration with NCBI in USA and EBI in Europe. So this particular database contains protein sequences and structures along with the nucleotide or genomic resources. So this is the interface of this particular database. By tomorrow, we'll also be exploring the database and how best we can retrieve sequences from it. So now we have the SwissPort. So now SwissPort is a protein database that also has other that has other databases or that about uh, different tools that can be used to analyze protein sequences. Because by the time we explore SwissPort tomorrow, we have a part of it that harbors that helps to store protein data sequences. We have some part that helps to model the 3D structures of protein. We have some part that helps us to analyze pathways pathways in which a particular protein has been degraded, and also we have different tools on this particular database itself. So now, SwissPort is a curated protein sequence database which strives to provide a high level of annotation, such as the description of the function of proteins, its domain, structures, post-translational modification, variant, and the likes. So, and this particular database now, it has a minimal level of redundancy and a high level of integration with other databases. Now, SwissPort now is integrated with several like Uniport. Uniport now is a database that is incorporated on the SwissPort where protein data are stored. So now tomorrow now we are going to major more on the Uniport database. So we'll just give an overview just for us to understand what are present on the SwissPort, and from there we maneuver our way and navigate our path into the Uniport database where we're going to retrieve one or two protein sequences. And this particular database now was created at the Department of Medical Biochemistry of the University of Geneva. So this is just the interface of the Uniport KB, where we are going to be retrieving our protein sequences tomorrow. God helping us. So now we also have now from there now we move to the secondary database. So now what are the secondary databases? These are databases that store the information derived from the analysis of primary data sets. So now when you as a researcher now you worked on a data that was retrieved from online database now. So now such data that you generated from your analysis now are termed to be a secondary data. And this secondary data now will now have a particular specialized database that are basically meant to help store those secondary data that you generated. So this secondary data now that you generated now might be informations that are gathered from the analysis of those primary data sets, just to help to give understanding into some to give meaning. Sorry, to times it might probably not be structures, so it might be information. Maybe you in the process you try to like get understanding to the function of a particular protein. Maybe you try to like classify the protein into different families or different domains based on their similarities and their differences. That is just what secondary databases now help us to achieve. Also, this database now contains highly curated informations which are derived from complex computational as well as manual analysis of primary resources and scientific literature. So most times it might be that it might not be the researchers who carry the analysis, the research work now that submitted the data online. Several of these secondary data now are generated, are retrieved or are gotten from scientific literatures which are published. So that is just one of the ways by which the management of secondary database now they help store some of those informations, and they help up to properly manage and help us to categorize them to different groups, so that by the time we want to access some of these data now, we won't have difficulty in navigating or in getting them. So that's just it. Also, this database is better and contain more valuable knowledge compared to the primary databases. Now we have more information in regards to a particular data set on the second database compared to the primary database. So now if by chance now, maybe you okay now, by tomorrow, maybe for example, we have a particular department database on the NCBI, we call it the Sequence Read Archive, SRA. So basically, if you want to work on whole genome sequences of an organism that has already been stored online, Sequence Read Archive or Bioproject, Biosamples, these are some of the databases that you can access to get to retrieve your data and compare it. So now, by the time you click on each of the data, we are going to see little information as to the state of the disease patient, the name, the sources, the source in which the data was generated from, and several others. This is just a few of the information we are going to have on the permanent database. But by the time this particular data now has been worked on by other scientists, then we can now have more information on that particular permanent database. So this is what second database helps us to achieve. So we have more and better information, valuable information now, or knowledge in regard to a particular data, permanent data that has been that was generated. And also, second database now often store information about conserved domain structures or sequences, signal sequences, and active site residue, and several others. Next time, maybe motifs and several others. So it helps us to give meanings to some of those of these. Also, now what are the examples? We have ProSite, we have the PFAM, we have the Scope, and the CAT. So now, ProSite, for example, now this is a protein database, and it consists of entries which describe the protein families, domains, functional sites, as well as amino acid proteins and profiles in them. And this particular database now they are manually created by the team of the Swiss Institute of Bioinformatics and tightly integrated into the Swiss Prot protein annotation. So this is just and one thing I forgot to make mention was that each of the examples we have under the secondary database now has their different area of specialization or their functions. So some, say for example, the process now, what is help us to describe protein families, their domains, functional sites, and the patterns of the amino acid that is present in them. That is just the area of specialization on this particular database itself. So now, if you now look, this is just the interface of this particular database, the PDB, the ProSite database. So now, following this, we also have the CATH, which is the CAT. So now, this particular database now is just an acronym of it's just an acronym of the criteria used in categorizing your protein into different domains. So now, and this particular database now is a protein structural classification base. This particular database classifies the structures of proteins in comparison to what we have earlier on the ProSite database, which helps to describe it and several others. But for this one now, basically it deals with structures of protein, and this particular database now is free and it's available online, and can help us to provide information on the evolutionary relationship that exists among protein domains. That is just what it helps us to achieve. And these particular domains now are classified into within the the CAT structural hierarchy. So the C stands for the class level, the A on the acronym stands for the architecture level in which is used to classify proteins into that domain. Also, we have the T, which stands for topology or the folding, the mode of folding of the proteins. And also the last letter, which is the H now, also stands for the homologous superfamily level. So this is just the criteria, the criteria that is being used. This acronym now the criteria forms the criteria that is being used in categorizing your protein into domains, and help us to it provides information, evolutionary information in regard to the evolutionary relationship that cut across structures of proteins. Also, this is the interface of this particular separate database. And also, we have the last database we have here now. We have the Scope, which is which stands for structural classification of protein database, and is a largely manual classification of protein structure domains, which based on the similarities of their structures and amino acid sequences. This particular database now, what it does, it helps us to give meanings to structures of proteins based on the similarities that occurs among the structures and the amino acid that make up that particular protein. So this is just what this particular database now help us to achieve. And also, it provides a classification of individual structural domains of proteins rather than classification of the entire protein, which may include a significant number of several different proteins. So now this is just the interface of the school database. So we are going to be exploring this tomorrow also. So now we also have two other databases now that are totally different from the two databases I've already mentioned, the primary and secondary database. Now we also have the composite database, and these are databases now that amalgamate different primary sources, often using criteria that determines the priority of inclusions of the sources and the level of redundancy are still retained. This particular database now, one of the examples of this database is the Unipost database, which helps to get data from two different databases now. It gets database from the Swissport and also from the Tremble. So by the time we are navigating our path through the Unipost database tomorrow, by the time we are trying to have access to information in this particular line, then we get to see how this particular database is incorporated into other databases in which they get their information from. So most times you will click on a particular link that's going to take you back to the NCBI database, or some taking you back to the EMBO EBA database, or even some taking you back to the Swissport and several other databases. So now there's a particular criteria of inclusion that is being used in sourcing out for data on this particular database, this particular type of database, computer database, just to help to reduce redundancies of data, and to make sure that the information that are present in each of the data are still retained and maintained. So that is just it. And the last one is specialized database. These are databases that are particularly or that cater for a particular set research interest. So now today we have different researches, different areas of research, and we've now created different databases now in that line. So today we have database that helps to house or to archive data, HIV viruses. We have some database that help us to archive SARS-CoV-2, Marburg, monkeypox, influenza viruses data. So example of databases that are specialized in storage of SARS-CoV-2, monkeypox, and influenza viruses database data now are the GISID database. And also, we have one database. Today we have different databases that have us plant data, different plant data. So some of them are specialized on herbs. Some of them specialize on vegetables and several others. And also, even within the vertebrate animals, we also have different databases that specializes in storage of data of different vertebrate animals. So today now we have different specialized databases on the basis of what the research interest. So now this will be the end of today's class. So by tomorrow we're going to be doing a little hands-on on some of these databases. If my partner not be able to touch all the database, at least the major ones, we are going to have access to some of the major databases tomorrow. So I don't know, maybe you have one or two questions in regard to what I've been said so far. So also by tomorrow we also probably do multiple sequence alignment, and likes, if possible, blasting. So thank you very much.
Yeah, I think we really appreciate your class. We just have like 3 minutes left to take the questions. By the time we are preparing for the second class now, which start by 11:30, which is for immunoinformatics. So you want to believe that It's going to be helpful. The class ends tomorrow after the practical. So during the course of the after the end of the class tomorrow, for those who have indicated in the certificates, we'll be sending the certificate to them immediately by tomorrow. The deadline for payment is tomorrow immediately after the class. Then the recordings will be sent and other material to support the field training we have done will actually be sent tomorrow. So Mr. Daniel or Dr. or Professor Daniel, if you have registered accordingly, you ought to have got to the link for the payment. Or if you have your friend that have registered, please just use that same link the person has. That would be very good. Because the way we are doing it is automatic and we don't want to be somebody that is affecting others. So once everything closes, we don't go back again to that. Like there are some people that were victim of clinical informatics, bioinformatics rather. It's thought we're going to extend it, but after that day, that's the end of it. It's not for profit making. So we have schedule of work to do. We have to go by the deadline. So tomorrow, after tomorrow, that's all. And it's better you go through Paystar so that the data can be harvested and the result, the certificate can be processed. In the past trainings, we having problem with certificates, or people paid no certificates. So this time around, we are putting a system in place. Mr. Bolu, can you mute your microphone, please? It's giving us echo. So it's really going to do that way. So for the next two minutes we have, Mr. Daniel, can you please ask the question? I see raising up your hand, Mr. Daniel Orike, or professor or doctor. Can you ask a question, please? Yeah. Please, can you ask a question? I think our HR have responded. I will not see email and you didn't want to pay, you can make a request to the HR at helixbiogen.org, and from there they'll be able to pick it up. You're going to have access to the video and the materials and your certificates. So any question from the participants? Daniel Orike?
Yeah. Can you hear me?
I can hear you.
Okay. Yeah, thank you very much. I'm so grateful for such great information we've accessed today. This is an eye-opener for some of us who are trying to explore this very platform, the bioinformatics platform. Well, my question was just on the aspect of the slides. I was about to ask, are these slides going to be available for us after now? Thank you.
Yeah, this is part of the material we send it with the certificate.
All right. Thank you. Thank you.
Yeah. In the absence of no question, okay, Dr. Professor Mr. Folawiyo Suleiman.
So he was mentioning about the type of databases, and
yeah,
he said something about accessibility, and I feel he did not explain anything about that. I guess
accessibility.
Yeah, he said the three types of biological database, that the categories you have: nature, the source, and he mentioned accessibility. So he didn't mention anything about accessibility, like at the end of the whole lecture.
Yeah, what we meant about accessibility is that not all databases are accessible. Yeah, but we'll be dealing with the ones that is open source, that is accessible for us by everyone. I can tell you categorically that I don't have access to Chinese database. It is the way they coded their database. So the few ones that you have access to, that is what we're going to teach you. And even the bioinformatics tools too, there are ones that is not open source that you cannot use unless you make payments. But we expose students to open source bioinformatics tools. There are some other organizations that do training that when they do training based on the tool they have developed. That even if you don't subscribe to it, you can use it. But what we do on our own hand here, we expose students to open source bioinformatics tools that anywhere you have in the world, you have access to it. You can use this. They have been verified. They have been tested. So, do we have any other question again?
Can you hear me?
Yeah, have I answered your question?
Yeah, I made just notes of 3 questions actually.
Okay, can you ask a question?
So I said, what does search query? What does it really mean in terms
What?
of search query? Search query. What does it really mean in
Search
terms of?
query. Yeah, it's going to do that tomorrow. It's Anton.
Okay.
It's Anton.
So, but
So it's going to do
the
that.
last question I have is that's just like a suggestion. Like I want to know how influential bioinformatics is with forensics. How influential are those two fields?
Yeah, they are
Like how
interrelated. They are interrelated. Because you take your sample, you sequence, you related. Analyze them.
Oh, wow. Thank you.
Yeah. We use it for crime detection and every other thing like that.
Which is something I'll be likely to look into.
Yeah. In absence of no question, we are preparing for the second class by 11:30. I think we're going to close this class. See you tomorrow by 10 a.m. that we do the proper hands-on. If your friend could not join today, you know him or her, kindly remind him or her that tomorrow is another good day. 10 to 12, a long hour tomorrow. Like this morning, I look as if we not even spent an hour. It's more than how we are spent now. It look as if we should continue. So tomorrow, 10 a.m. West African time, tomorrow, Lagos time, we join this same link. No changing. We are still here. See you tomorrow. God bless you. Thanks for joining.